All right, guys, going through AS Biology paper one. This is the 2024 paper. Just quick mic check there. Hour and a half minutes. What we got? One, two, three, four, five, six, seven, eight, nine questions. So, this is the 2024. What I recommend is you press pause on the video at the start of each question, get a piece of paper and a pen and scribble down your answer. And then check your answer off against my walkthrough by unpausing the video and watching the explanation. Don't want to say that every time, so I just recommend at the start of each question you pause, scribble down, and have a go. I've kind of lost my editing software at the moment, so I'm doing a few with my face on so I can tell you when to pause until I get some new editing software where I can add in my little bits like the little pause symbol and all of that. All right. Figure one. Question 1.1, figure one shows a diagram of the human gas exchange system. Here's figure one. We've got A and B. Surprise, surprise. I wonder what they're going to ask us. Okay. The structure B is muscle. Ooh. Structure B is muscle. The volume of the thorax is increased when structure B contracts. Name structures A and B. Okay. So, Zing Zang Zong, A over here, that is pointing to, well, the growth structure here, this big tube here running down your throat. This is the trachea, your windpipe, known as the trachea. It's really hard to see whether they want you to actually just talk about one of the rings of cartilage that strengthen and support the trachea. So I think they'll accept both. It'll either be trachea or ring of cartilage. I'll go with trachea. And then B is an intercostal muscle. Now, when we breathe in, our external intercostal muscles contract. When we breathe out, they relax and the internal intercostal muscles contract. Okay. Or certainly when we do forced expiration, the internal intercostal muscles contract. But this is going to be external. So my answers are going to be trachea, although they will accept ring of cartilage. I don't know how to spell cartilage, right? I'm always worried. Is it D? Oh, it's definitely not cartilage. Carti. That's how I'd spell it, but I just think the D looks a bit weird. Okay, let's leave that out. And this will be external intercostal muscles. Maybe that's one for a revision card. The internal and external intercostal muscles when your ribs move up, when you breathe in, or when your ribs move down, or when you're doing forced expiration. Maybe that's one for a revision card because they're hard to memorize. Um, okay, 2 marks. Let's move on. Otherwise, we're going to be here all day and all night. My God, my face is taking up far too much of this screen. Let's zing down. Oh my good gosh. Let's move that out of the way so it's not covering up the screen. 1.2, figure two is a diagram of an alveolus. That's a singular instead of alveoli, and a blood vessel associated with the alveolus. So we've got a blood vessel wrapped around this one alveolus. The direction of the movement of substances is shown by the arrows labeled C, D, E, F, and G. So a substance is coming into the blood vessel, and then we've got a substance leaving the blood vessel. Then we've got a substance D coming in and out of this alveolus. And we've got a substance crossing from the plasma of the blood into the alveolus, and then a substance crossing from the alveolus into the plasma. So we're probably going to be asked what these are. Okay, yeah, so we've got to do 3 of these. Complete table 1 by writing the correct letter C, D, E, F, or G in the box next to each statement. You may use each letter once, more than once, or not at all. So you need to put like one of these letters in each of these boxes. And you could use the same letter in more than one if you think that's right. So which one represents the diffusion of oxygen? Which one represents the transport of a gas mixture? And which one represents blood moving at the highest pressure? So I'm going to go through the answers. You might want to pause, have a go. All right, let's go. So I'll go through all of them. So C, we've got the blood entering the capillary, and this capillary is wrapped around this alveolus. And we've got the blood leaving the capillary here. Which one represents the diffusion of oxygen? So that's blood, C and E, so that's not oxygen. G, what's going to leave the blood and go into the alveolus? That's going to be carbon dioxide because we breathe out carbon dioxide. F is going to be oxygen diffusing from a high concentration to a low into the blood plasma, then into those erythrocytes. So F is going to be oxygen. Our first box would be F. What is the next statement? Because my crappy brain has already forgotten. What one represents a gas mixture? So that's going to be air. D is going to be air. So it's going to be a mixture of carbon dioxide, nitrogen, oxygen. That's going to be air going in and out. And then the final one represents blood moving at the highest pressure. So most of you will know, well, it's the blood in C and E. So is it at a higher pressure after or before? So you got to think, where does the blood come from in the capillary that goes around here? It comes from your pulmonary, here's my crusty little heart, and you've got your pulmonary artery that's connected to the right ventricle, and the blood is going from the artery into an arteriole and then into the capillary. So we're looking at the arterial end of the capillary. That's going to be the venial end, and it's going to connect to the pulmonary vein and take it back into the left atrium from over there. So our highest pressure is going to be here at C because it's just come from that contraction of the right ventricle. Okay, Coolio. So can I remember the order? So the correct order, peeps, is diffusion of oxygen was F, mixture was D, and then represents the blood at the highest pressure will be the arterial end, the start of the capillary, and that is going to be C. And that's worth 2 marks. Now, it won't always be on the left, the arterial end of the capillary. So just take into account the question and you can kind of work out what's going down. All right, which direction it's flowing. They gave us arrows, so we knew which the arterial end was. Right, let's get rid of that and stop waffling. Let's move on. Is that the end of the first question? Was it just a 5 marker? No, it's a 6 marker. Okay. Describe and explain one adaptation of a single alveolus for gas exchange. Now, this one, I reckon quite a few people may have made a mistake on this one. All right. And the reason is because it specifically, you have to pay attention to the bold black, especially in A level, okay, and in GCSE, but that means for a single alveolus. Now, we might want to say, so the misconception here, you might want to pause the video, grab pen, paper, have a go at this one. All right, I'll go through it. So the misconception here is that when we look at an alveolus, they've given us a single alveolus looking like that. We might be thinking of that drawing. That is not a single alveolus. That is kind of showing like multiple alveoli. And when you've got multiple like that, the way they're arranged, they're going to have a large surface area for a rapid rate of diffusion. However, we're looking at a single alveolus, so we can't really say, oh, it's got a large surface area. There's nothing highly folded about that single alveolus. So don't go down the surface area route. They're not going to give you marks for that, although, you know, you're not totally incorrect. So we want to go down the thin walls. So we're going to say, now you can say thin walls. I'm going to say of the alveolus. And these are going to be the walls of the epithelium of the alveolus. I don't know whether they're going to definitely demand that. It's going to be the squamous epithelium. Squamous are the squash cells. I don't think we're going to need that at AS, squamous, but epithelium cells. Oh my gosh, sounds terrible. Right, so thin walls of the alveolus. That is the description. So that is 1 mark. Now the second mark, so we've done the describe. What's the explain? How do thin walls help gas exchange? Okay, they make a short diffusion distance. A shirt. Bloody hell. Lucky I didn't do that without the R. So that definitely would be incorrect. Short diffusion distance. I don't know what we're going to say for rapid diffusion. Okay, you can probably just say short diffusion distance. You don't have to say for rapid diffusion. So thin walls, or you can say squamous epithelial cells, which means they're squashed, they're flat, or flattened epithelial or flattened squamous cells. I always think of the SQUA as like the word squash when you spell squash. So it's like they're squashed cells. Short diffusion, blah, 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 blah. That's your 2 marks. All right, I'll stop waffling. Right, let's go on. And what I was going to say is you don't have to say the alveolus. We could talk about the thin, 1 cell thick walls of the capillary. I might want to put that this is one cell thick as well. Both the alveolus and the capillary are one cell thick. So if I was in an exam, I'd probably bust out they're one cell thick there, just in case, because you never know when they're going to do that. Oh, you didn't say they're one cell thick. You know, you don't know. I'm sounding a bit thick right now. Right, let's go to table 2 and 2.1. So, table 2 contains information on the digestion of two biological molecules. Complete table 2 is 4 marks. Here's table 2. Biological molecule, we got starch. They want us to give the name of the enzyme, obviously, that breaks starch down, the name of the bond that it hydrolyzes. Hydrolyze means breaks using a water molecule. And the product of the digestion they've done for us is maltose. So that's going to get us 2 marks. And then the second one is the biological molecule is a dipeptide. They've given us the enzyme is dipeptidase, and they want which bond would you find in a dipeptide that needs to be broken, and what would the products of breaking down a dipeptide be? Pause, scribble your answers. Let's go. All right, so the answers are starch is amylase. Now, at GCSE, it's always weird because you tell students lipase breaks down lipids, protease breaks down protein, and then you say amylase breaks down starch. And you look at the names and you're like, what link do they have? Of course, starch is made of amylopectin and amylose. So amylase, it makes more sense at GCSE if we called starch amylose or amylopectin, kind of that would help realize what you do with ends. Anyway, enough whiffle. The bond, you know it, it's glycosidic. So glycosidic bonds, that's what's hydrolyzed. And that's going to get us 2 lovely jubbly marks. Now we're going to move down here to the dipeptide. Now the dipeptidase is going to hydrolyze the bond in a peptide. That's a polypeptide or a protein. This is just a dipeptide, 2 stuck together. So it's going to be a peptide bond. And the product, when you break down a peptide bond, you're going to release 2 amino acids. Hopefully we don't have to say 2. It's just amino acids, right? I hope so. There we go. Bish, bash. Okay, let's move on. A student investigated the digestion of starch by mixing starch with a solution of the enzyme used to digest starch. They don't want to give us the name, although we know it's amylase. The student did a biochemical test for protein when starch digestion was completed. I can't speak. Describe a biochemical test to show the presence of protein, 2 marks. So this is nice. It's just name, color change. So we're going to go with, and you don't have to warm this one. It's Biuret's reagent, which is kind of a mixture of like, oh yeah, we don't need to know. So let's not talk about Biuret. All you need to know for AQA is just the name. So Biuret's reagent, mix the food sample with the Biuret's reagent, or mix the sample with Biuret's reagent. It goes purple. Now you don't normally have to state the color before. The way I know that it goes purple, I think of the P in protein stands for pink or purple. Now you can say lilac, mauve, or violet. They're all possible answers, but they don't begin with P. So I like to stick with pink and purple protein. So it kind of triggers in my head which one it is. And Biuret's begins with a B. It's blue. And when you do Benedict's solution, that also begins with a B. They start off blue, but both of them change color. And they're not normally interested in what color they are at the start. Okay, I can't think of them ever asking that. Okay, so it's Biuret's goes purple. Bosh. Oh my gosh. Where are we? 2.3. The student's test for protein was positive. Now they only had amylase and starch, or amylase and starch. Explain why the test for protein was positive. Hold on. We didn't put a protein sample in there. We put starch in there and then the enzyme that digests starch. Pause the video, think about it, have a go. All right, I'm going to go through the answer. So the answer, of course, amylase is an enzyme and enzymes are protein. So we want to say amylase. Oh my gosh, I've got some fat fingers. Amylase is, and well, yeah, the rest of me is fat as well, unfortunately. Amylase is an enzyme and they are made of protein. So there we go. Now, when I first had a go at this paper before, when it first came out last year, 2024, I was like, and they bear made of protein. Oh, whatever. Okay, I thought that would be enough for 2 marks because you're saying, oh yeah, amylase is an enzyme and enzymes are protein, 2 marks, but it's not. What they want you to say is, and all the starch is digested, but the enzyme is not broken down. The enzyme remains. So basically, enzymes are catalysts. I can't spell, and so are not used up. I can actually spell, it's my typing that's messed up. So enzymes are catalysts and are so not used up. So the enzyme is still present. The protein hasn't been broken down, which is why we get the positive result. So we kind of needed to say that, which, yeah, I think that was a difficult mark. It's a potential drop of a very easy mark there. Okay, let's smash down here. So don't beat yourself up too much. They didn't make it clear. 2.4, an enzyme's turnover number, kcat. And this is like an application question. They're giving you a maths question, but they're throwing in some context that you're unfamiliar. They're putting it in unfamiliar context. You probably won't have heard of kcat before. An enzyme's turnover number, kcat, is the number of substrate molecules converted into product molecules by one enzyme molecule in 1 second. It is determined using this equation. Kcat equals the maximum rate of enzyme-controlled reaction in millimole per decimeter per second over the enzyme concentration in, micromoles, not millimoles, micromoles per decimeter. A scientist investigated the action of the protease enzyme. The scientist prepared a reaction mixture with a protease concentration of 0.0118. The kcat for the protease is 110 substrate molecules per second. Use this information and the formula to calculate the maximum rate of protease controlled reaction. Give your answer to 3 sig. So what I want to identify is all the important parts of this question. So this is where I'd say bust out a highlighter and then pause the video, have a go on a bit of paper, get your scientific calculator out, and then I'll go through. Okay, so if we're busting out the things to highlight, obviously they've given us this and we're like, oh, what is that? That's enzyme conc, right? They've given us the enzyme conc because it says protease conc and protease is an enzyme. They've then given us how many substrate molecules per second. That's the kcat. So they've given us that. And then we need to also pay attention that it's 3 sig. So here's all of our marks in these key bits. And then we can put that in a triangle if you love up triangles. And whatever's at the top of the divide line goes at the top. So this is the thing we don't know, the max rate of enzyme controlled reaction. And that is what they want us to calculate, maximum rate of the protease controlled reaction or enzyme controlled reaction. And we know that. So it's kind of that at the top. And then you've got kcat and enzyme conc at the bottom. Oh my days. So we're going to times those together, and Bob's your uncle, Jill's your aunt, and we're in there, right? So we're going to go 1110, that's the kcat, times 0.0118. I'm going to bust that in the calc. What is that? 0.0. Oh my gosh, this is where my fat fingers and my calculator skills are bad. 1.298. So I reckon you're going to get 1 mark for this, maybe 2 marks for this, 1.298. So just realizing where these go was key. And that's going to give you 2 mark. No, it's going to give you 1 mark because, of course, we need 3 sig. So that's going to give us 1 mark. Thought it was being a bit generous. And then we need to 3 sig this. So the 9 is bigger than 5. So we're going to round that up, and that's going to push that up to 3. So we're going to end up with 1.3, and then we go 0, and that's 3 significant figures. So 1.30, and that's going to be in micromole decimeters per second. Awesome. Let's zing on. We're on to question 3. Describe how viruses are replicated. Now, this is quite a common, this is a revision card question. So bust out a, oh my gosh, bust out like a revision card. If you don't know this stuff, when you go through the videos, bust out revision cards and make them. If you don't know stuff, put it on a revision card. You don't have to do the whole course on a revision card. Why are you going to put things that you already have locked down from GCSE and in AS that you know off by heart? Just put things you don't know down. So if you have trouble with this one, this has come up quite a few times in A-level biology, and it's just a 4 bullet point marker how the viruses reproduce. So I'd put like the question there and then the answers on the back. You can actually get like 2 questions about viruses and 2 sets of answers. But there we go. There's a good way to just get those memory recall. And remember, can't even speak. Biology is a huge, huge memory recall topic. Right, that's nice low grade boundaries. Right, describe how viruses are replicated. So remember our virus, however our thingy virus looks, whatever, let's not draw That's some dodgy virus. Anyway, what the hell is that? I did get a level 4 in GCSE art. So this is an attachment protein, supposed to be, and then that's going to bind to like a protein on the cell membrane. It's going to lock into a protein receptor site on the cell membrane of a cell, and then it's going to inject its like genetic information, we need to be more specific there, into the cytoplasm of the cell, and then it's going to take over the transcription factors, and we're gonna it's going to reassemble lots of viruses and burst out the cell. Let's put that into words properly. So we want to say, for one mark, the virus attachment. Oh my days. See, I don't have my editing software at the moment because otherwise what I do is I just go through and cut out all my stupid mistakes when I'm spelling and stuff to like make it all a bit slicker, but I don't have that at the moment. So we're just going rough and ready. The virus attachment protein binds to receptor site or receptor protein on host cell membrane, cell surface membrane, whatever you want to say, right? So that's definitely going to be a mark. Then what happens? The virus injects into the cell. So the virus injects into the cell its genetic material, but at A level, we've got to be more specific. So I'm going to say, you could say it's capsid because the capsid is going to contain, no, I'm going to go with injects into the cell its DNA or RNA. You could just say nucleotides or nucleic acids. No, you can't say nucleotides because that'd be all broken up. Viruses are either DNA viruses or RNA, their genetic material. So I would go with DNA or RNA. And then what we want to say, the enzyme that is already in human cells, this is it's not talking of specific in a human situation, could be an animal getting infected with a virus, could be a plant, could be anything. So we're just going to say reverse transcriptase enzyme. Okay, turns RNA turns RNA into double stranded. So it basically, if it's an RNA virus, it turns the RNA into a double stranded piece that can now be nicely connected into the host DNA. This is inserted into host cell DNA, transcribed. So it gets transcribed by DNA polymerase in the host cell to form viral proteins. So, yeah, there's probably like 4 marking points here. Virus injects the reverse transcriptase, and then maybe that's and inserted into the host cell DNA or and transcribed. No, transcribed to form viral proteins is a separate mark. But also you get a separate mark sometimes for saying, but not always, for saying the viral proteins are then assembled. They're then like put together to make new viruses, to make new viruses. I don't like that sentence, but there we go. These burst out of the cell, and then they go and so the virus, they burst out, and then they go and infect other cells. Okay, but they didn't really ask about all of that, so yeah, there's like 5 marking points there at least. All right, let's move on. So I'll get that on a revision card just in case it comes up. Okay, 3.2. A scientist investigated the effect of a virus species on a bacterial culture. The scientists mixed a diluted sample of the virus with a bacterial culture in liquid agar, poured the liquid agar into a petri dish and let the agar set. Incubated the virus bacterial culture at 30 degrees for 24 hours. Information. Figure 3 shows the appearance of the petri dish after 24 hours. Excuse me while I have a drink of coffee. Wrong hand, sorry. Holding it like an ape. Figure and slurping away down your ears. Figure 3 shows the appearance of the petri dish after 24 hours. Yeah. So we got the cloudy agar with growing bacteria. You did this at GCSE, and you got these clear zones. I can't remember the halo of inhibition or the zone of inhibition, whatever they were calling at GCSE. Okay, explain why the clear zones are present in figure three. So what's happened to the bacteria in the clear zones? Well, they've been killed. So bacteria have been killed. And that's all I'm going to put, folks. They just want us to understand what's going on there. Replication has been inhibited. They're not replicating. The bacteria have also been killed. So there we go. Predict the appearance of the Petri dish if the incubation was extended for a few more hours. So they did this for 24 hours. So let's say they did it for 28 hours. What would we expect? Um, Now it's not antibiotics this time. It's a virus that kills the bacteria. So it's like a bacteriophage or whatever. It's like destroying the bacteria. So we would expect the white patches or the clear zones to get bigger, right? Because you're giving the virus more time to replicate, go out and destroy other bacteria, essentially. So that's all we need to say. We would expect to see the clear zone, the diameter of the clear zone should increase. Should be just a one marker, yes. Okay, the clear zones increase in size. Lovely, bubbly, jubbly, awesomeness. Let's go. Okay. Um, 3.4, animal cells replicate during the cell cycle. Figure 4 shows one animal cell at a stage of the cell cycle. So we're talking cell cycle. You should know this off by heart. You know your stages. You got your four stages. You got mitosis. Then we got, is it G1? And then S and then G2. Some break it down into G1, 2, and 3. I can't really remember. It depends on what syllabus you're doing. I can't remember for AQA. But yeah, you've got all your different stages. So we've got a stage of the cell cycle here. Can you recognize what stage that is? Looks like two fried eggs or two eyeballs, but it's not. Identify the stage of the cell cycle shown in figure 4. Explain the appearance of the cell shown in figure 4. Do not refer to organelles. So they've told us specifically don't refer to organelles. Again, this is the type of thing I would make a complete balls up of in the exam. So do not refer to organelles. I'll start bussing out about the nucleus and stuff like that, and I wouldn't get a mark because the nucleus is an organelle. So, yeah. What stage do you think this is? There's two options. I'm going to give you the answers. So identify the stage. We can go for telophase, which is the last stage in mitosis, or we could say cytokinesis. So pat yourself on the back if you got it right. Um, Cytokinesis is after mitosis. So if this question had said which stage of mitosis this is, it would be telophase or telophase. Um, but it just says cell cycle, so it could either be the telophase or cytokinesis because cytokinesis, they kind of blend into each other. And the explanation is we can say the cytoplasm is cleaving in two. So they're not really counting the cytoplasm as an organelle, as a separate cell structure. We could say the cytoplasm is dividing. Two daughter cells are forming. So two new daughter cells have formed, and I would add the cytoplasm is dividing. Now, it's tempting here to say, oh, nuclear membrane or new nuclei have formed, because that happens in telophase. Nuclear membrane reforms around each set of chromosomes, and we get two new nuclei reforming. But they told us specifically don't refer to organelles, so they're going to brute us for that. So really what we want to say is there are two daughter cells, and we can see the cytoplasm dividing. Maybe we could say the cytoplasm is pinching or budding. They're budding or pinching in the ends, like here and here. But there we go. Yeah, so watch out for that one. They weren't going to give us the marks if we talked about nucleus. Okay. 4.1, complete table 3 to show whether the feature of DNA is associated with the DNA molecule found in each of these locations. Tick the appropriate box. So what we got here, 3 marks, table 3, 3 rows or 3 columns. Let's have a look. Feature of DNA, it is circular. All right, so we need to put a tick. Which one of these has circular DNA? Why don't you pause the video, have a go at this one on some paper. All right, I'm going to go through the answers. Let's go. So which ones are circular? Well, prokaryotic cells, they have, you can all picture they got their little plasmids, but that's not circular DNA. They're tiny circles, just a few genes. Prokaryotes have a kind of loop of DNA, chromosomal DNA, that is circular. And guess what? So do chloroplasts. They have circular DNA. Is there circular DNA in the nucleus? No, our DNA is known as linear, and that means it's not joined up at the ends. So it is double helix. Oh my God, that's the worst double helix ever. But look, the ends are open, they're not joined together. So it's linear DNA for us in our nuclei. Contains four different types of nucleotides. Well, what do we know about DNA? We know it's non-overlapping, it's degenerate, it's universal. The same four nucleotides do the same thing in all life. So yes, we're going to find four nucleotides in bacteria, in the nucleus, and in the chloroplasts. Is associated with histones. So remember, our DNA is attached or wound around histones. It's more like wound around like that, and it helps the histones help condense the DNA, whereas circular DNA is all loosey goosey and open. This is all condensed around histones, and that is only going to be in DNA that's found in a nucleus. It helps pack lots of DNA into the nucleus. You're not going to find that in prokaryotic cells, and you're not going to find histones in the chloroplasts. They've got the circular DNA. Again, that's endosymbiosis. You want to check that out. It's not in your syllabus, but a bit of extra reading. Endosymbiosis is the kind of theory that in the past, chloroplasts and mitochondria were kind of like prokaryotic cells or some sort of relationship to prokaryotic cells, and they eventually came to live in eukaryotic cells, etc. Da 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 da. Um, Wonderful. That was 3 marks. Let's zing on. We're just going to finish up question 4. Some strains of the bacterium E. coli, I can't say that, Escherichia. Es- All right, let's leave that. E. coli are known to be resistant to antibiotics. E. coli is a big killer in the UK and all over the world, by the way, massive killer. Um, Scientists analyzed data recording the deaths of people in hospitals across the world during 2019. They used the data to find how many of the bodies of people who died in hospitals contained antibiotic resistant E. coli bacteria and whether the E. coli were the cause of them dying or not the cause of death. If they weren't the cause of death, they were called associated E. coli. They were just in their body, probably a correlation. But if they actually caused the death of the patient, they were known as causal E. coli. And here are the results. So we've got associated, lots of people found, so that's sort of 0.7 million, whatever that is, 700,000 people kind of died with E. coli in their body, but it wasn't the killer. And then it looks like about 1.8, I don't know, 180,000 people sort of died, and E. coli was the cause, direct cause of their death. What's this? This is going to be a mathsy. Yeah, it's a maths question. 4.2, use figure 5. Using figure 5, a student calculated that out of all the people who died when antibiotic resistant E. coli were present, 25% of the deaths were causal E. coli. The student's calculation is incorrect. Use figure 5 to calculate the correct percentage of deaths involving causal E. coli in people who died when infected with the antibiotic E. coli. Identify the mathematical step the student performed incorrectly in their calculation, 3 marks. So we need to do the correct maths and work out the correct percentage of people who died because directly, causally, the E. coli killed them. And that's going to get us 2 marks. And then we need to identify what they screwed up where to get 25%. I'm going to go up to the data and do the question there rather than down here, folks. All right. So first thing to note here, you might want to pause the video and have a go. They're basically asking you, what is the percentage of the total people that died from E. coli? What is the percentage of causal? So I can already see the mistake the students made. So here, by the way, you can work this out, and that is, oh my good gosh. Well, we've got 10 squares and we've got zero 0.2. So it's like 0.2 divided by 10. Right. Yeah, whatever. These go up in 0.2. Going to divide it 0.2 by 10. Does that make sense? I don't even know what I'm talking about. Oh my God, I've got to switch my calculator on. So each square is one, oh my days, one square equals 0.02. Okay, Tom. Right, so we got like 0.62, 0.64, 0.66, 0.68, 7. zero point 0.7, 0.72. So I'm looking at 0.72 right there. And then here we're looking at, that's zero point 0.1. And then we're going up to 0.18 there. Now, I've already seen what the student has done as a mistake. So when we come to say what the mistake was, what is the mistake? And some people will have done this as their correct answer, and they'll get this, then they'll know, oh, sugar, this is what the student did. So they did 0.18 over 0.72 times 100. And if you do that, you're going to get, that should equal 25%, right? 0.72 times 100. Yeah, that equals 25%. So that's the mistake. The student divided 0.18 by 0.72, and that's all you need to put for that mark. So that's going to get you a mark. Now that's the last mark. The actual first two marks are do it correctly. So what did the student forget? The question asked for you to work out how many people died directly of E. coli of all the people infected with E. coli. So we need to add this and this together to get our total. So if we go 0.72 plus 0.18, that is going to give us, even I can do that mental maths, 0.90. So we're going to go 0.90 and we're going to go 0.18 divided by 0.90 times 100. Bish, bash, bosh. That's what coffee does to me. It makes me do all stupid kind of noises and stuff. 20%. So, that is the correct answer for 2 marks. Bish, bosh. And that was a 3 marker, I believe. So I've already forgotten whether it was or not. Excuse me, will our coffee break? I'll just mute so you don't have to hear me slurping. Okay. Uh, oh my days. Don't sit the screen, you Wally. Right. 4.3. Which method is most likely to be successful in decreasing the frequency of antibiotic resistant bacteria in populations of people? How can we lower the amount of E. coli bacteria in people? The frequency of the, and not the E. coli, of the antibiotic resistant E. coli. So when the people died, this was the E. coli found in their body, whether it killed them or whether it was just associated. Give people lower doses of antibiotics. Well, if you take less antibiotics, I see what they're getting at. They're trying to trick you there because like, oh, if people have less antibiotics, then maybe less resistant bacteria will beat up. Now, lower doses of antibiotics are going to encourage more resistant bacteria because you're only going to kill the really weedy ones that aren't resistant, and you're going to allow the resistant ones to survive. So, if anything, you want to increase the dose of the strength of the antibiotics. If you're giving someone antibiotics, make sure you kill the resistant ones. Otherwise, they'll survive by natural selection, pass on their gene, blah, blah, blah. So that one is a test more people to determine if they are infected with antibiotic resistant bacteria. Now, that's a great first step in decreasing the frequency of antibiotic resistant bacteria because if you can find out they're infected with it, yeah, but that's only really giving you information. It's not doing anything about the problem. So we're going to, which means this one has to be right. And vaccinate more people to reach herd immunity. Once you get like 70% of the population vaccinated, then if you have, if you are if I've got the E. coli bacteria, I don't know, and I don't wipe my hands after going to the toilet, and I shake your hand, you're resistant, so you won't get infected by it. So once we've achieved herd immunity, we will stop the spread of it. So yeah, that would be a possible vaccinate against it, and that will get us the lovely jubbly mark. That's a bit of a gross thing to talk about. Right. Yeah, we're going on to question 5. I should just shut up and get on. I can't wait to work out. The moment what's happening is I'm kind of dicing between 2 different softwares. So when I make my videos, I use 2 different video editing softwares, and my current one that I've been using for the last 3 years I've been doing my channel basically took away a lot of the and I can't draw on the screen after I've recorded the video, which is a pain in the ass. and it's taken away quite a lot of the editing options because it's not what people want. They're all making AI videos now. So they're like, look, customers don't use this, and this and this. So we're taking those tools away. And I'm like, but I pay you for it. So there we go. So I'm in the process of trying to find a decent bit of video editing software. And, I'm hand to mouth here. Like I don't make no money from these videos really. So basically, yeah, I can't afford to pay for some super duper expensive one unless it was part of my everyday job, which intuition it is not. So, all right, enough crying. Sorry, I'm a beater. Let's move on. Okay, where are we? Where are we? Where are we? It's so hot here, man. I am sweating. I'm just continuing on with this 2024 Paper 1 AS Biology, AQA. Took a short break there of a day or two. 5.1, Malva sylvestris is a plant species. A student wanted to determine the mean height of M. sylvestris, plants growing in a field. Describe how the student could obtain measurements of the plants and in bold, so we need to do this as well, then process these measurements to obtain a reliable mean height of plants growing in that field. So let's have a little look here, folks. We're going to do a field. Do you remember from GCSE, if you want to study a feature of plants in a field, you want to make sure that you randomly pick your sample um, to avoid bias. How do we do that? We're going to use quadrats. We want to make sure we describe the process by how we randomly place the quadrats. You might remember it was one of the core practicals in GCSE biology. So what I'm going to do, I'm just going to say randomly place, I don't know, 10. I think 10 quadrats. 10 is a minimum sample at A level. So maybe we'll go 15 just in case. Just in case what? If 10's minimum, 10's minimum. Randomly place 15 quadrats in the field. Now, was it one field or two fields? Add a student measurements of plants and then process these measurements reliable means growing in a field. Yeah, okay, in that field or in a field. Okay, right. Randomly place 15 quadrats in the field. Let's explain how to do this by drawing ooh, a map of the field. So we want to draw a map of the field and dividing into a grid. So essentially, you've got your map of the field. We want to divide this into a grid. This is the most crusty grid ever, but we're doing it all for speed, right? And then you're going to number the grid. You might do coordinates. I'm just numbering it like this. Um, yeah. Okay. We get the idea, Tom. Okay, so we've numbered it now. We've divided it into a grid. Now what we want to do is say how we're going to randomize the selection of that number. So you could say, write the numbers on a bit of paper, jumble them up, put them in a hat, but that's long. Let's just say use a random number generator on a calculator. Yeah, use a random number generator on a calculator to select 15 numbers at random, place quadrat on that square. Now we want to standardize the measurement. We're measuring the height of the plants. We want to standardize the measurement, a reliable mean height of plants growing in that field. So place quadrat on that square. Let's go measure from soil level upwards with measuring tape, I don't know, in centimeters. Do the same measurement style, technique, whatever, keeping everything standardized basically for each plant. Um, calculate mean height for field. What else do we want to do? I mean, I think we've nearly got it all. They did say the word reliable over here to obtain a reliable mean height. So we're going to need to say we're going to repeat it because to get reliability, you're going to repeat. So repeat, I don't know, 3 times and calculate the mean height. There we go. Hopefully that's good enough. I don't know whether we should have said anything else. And then the process these measurements to obtain a reliable mean height. Yeah, process the measurements, repeat 3 times, add together all the measurements, divide by 15, whether we've actually got to say that. But yeah, calculate the mean. That's processing the measurements. I don't know whether we want to say remove anomalies if we compare, but yeah, whatever. Let's leave it at that. That sounds like 5 marks to me. Hit it up in the comments if I missed something. It's always good to have someone say something nice like, "You donkey, you just read out the mark scheme." I couldn't be bothered to look at the mark scheme, so not for that one. Right, 5.2. The student wanted to present the results for the distribution of plant heights in a histogram. Figure 6 shows the outline of a results table. Use figure 6 to design a suitable results table. The student should use for a histogram of plant height measurements ranging between 60 and 120. So what is a histogram? A histogram is like a bar chart. And what you do with the histogram is, depending on the range, you increase the width of the bars. So you have the height, whatever this is going to be, that present the results for the distribution of plant heights. So you'd have like 60 to 70 centimeters. How many plants were in that range? So number of plants in 60 to 70, number of plants in 100 to 110. And so the width of the bars should be equal to the range of that bar. Anywho, enough of my whiffle waffle. Let's get on and write the things in. So your independent variable, what you change, always goes in the first column of a table, and your dependent variable, what you're actually measuring, is going to go in the second column. Let's make sure I've got all of these in. Yeah, they're all in. All right, shows the outline. Use figure 6. Oh my God, I've missed what the titles are. Student, let's shrink this. The student wanted to present the results for the distribution of plant heights in a histogram. And what we want to do is, shows the outline, design a suitable table used for a histogram of plant height measurements ranging between 60 and 70. So let's do that. Here is, we're going to go plant heights, because that's what, yeah, we're going to go plant heights in centimeters. And yeah, I'm doing it in centimeters as they use centimeters here. You could say millimeters, meters, whatever you want to measure it in, inches, feet, whatever. Plant heights in centimeters, and then we want number of plants. This is really plant height. Oh my gosh, range, plant height range. Let's call it range in centimeter. Okay, so we've got 60 to 120. So what we want to do is fill this first column up. That's going to get you a mark. Hopefully they're good enough. Maybe they want frequency or something here, or frequency density, but I'm going with nice and simple, number of plants, plant height range. So with a histogram, you're looking at the width of the bar is equal to the range, and the number of plants would be going up on the y-axis, and the range would be across the bottom. So we're going to go 60 to 69 centimeters as our first. Then we're going to go 70. Notice I've got no overlap between the ranges. I've not gone 60 to 69, 69 to whatever. Yeah, I'm not overlapping. So every centimeter in between 60 and 120 I'm using. So then that's going to be these. Then I'm going to go, no, I was about to go 79, but we've already used that there. So we're going to go 80 to 89. Come on, Tom, speed it up, mate. 90 to 99. Yeah, that's going to work out perfect. Is it? Maybe not. And then, oi, oh my good gosh. 100 to 109. It's not really worked out perfect, has it? And then what do we want to do? 110. And we're going to go to 120. How did that work out? 90 to 99. Yeah, these are all going up in nines. And then my last bar, freakily, is 10 centimeters. Well, yeah, there we go. The width of the bar when I plot it out. Hopefully we haven't got to plot it out, but on a graph paper, slightly wider here for this because the range is more. And then the number of plants, well, we don't know that yet because we didn't carry out the study. Okay, let's go. That'd be like the second mark. Hopefully, I better check the mark scheme on that, but I will check later. All right, let's move on. Hopefully I got 2 marks there. Otherwise, it's all pointless, isn't it, really? Right, 5.3. A student investigated biodiversity in a community of bee species in a field of wild flowers. The student trapped and identified the bee species in the field over 6 months. Table 4 shows some of the students' results. Bee species, we've got the cuckoo bee, the domestic honey bee, the clover bee. Total number of trapped bees after 6 months. So they trapped 250 cuckoo bees, 800 domestic honey bees, and 200 clover bees. Okay, right. Awesome. The index of diversity in a community of organisms is determined by using the formula little d equals big N, n minus 1. So that is the total number of organisms. So that's all the organisms times all the organisms minus 1. And we're going to divide that by the sum. This little squiggle means the sum of the organisms of each species times their number minus 1. So 250 times 249, 800 times 799, 200 times 199. And adding that all together, that will be the bottom bit. Okay. What is it? Denominator and nominator? I can't remember which ones. Use the formula and the information in table 4 to calculate the index of diversity in this community of bee species. All right, let's do it. Let's do it. I'm going to do it over here. All right, then. Right. So, first of all, let's work out N. So we're going to go 800 plus 250. That's 1,050, 1,250. So we know that N is 1,250. That's N. And so I'm putting that up the top there. And what we want to do is times that by N minus 1. So nice and easy, right? Times 1, 2, 4, 9. And that is going to be at the top. I'm going to smash, where's my calc? I'm going to smash that in the calculator. 1250 times 1249. And that equals 1561250. 15. Oh my gosh, I'm gone blind. 61250. 61250. Honestly, I got these glasses from the charity shop and they were £2. Check them out. They got like a camouflage safari. I think they're for a 60-year-old female librarian, but they're good for me, right? They're £2. So, you know, like I say, bread and water here. 1561250, and I'm going to divide that by the sum of over here, this total number of organisms of each species. Oh my gosh. So I've got to do 250 times 249. This is for the bottom bit that's going down there, right? We're doing that down there. 800 times 799 and 200 times 199. And we're going to add that all together. I'm bound to make a mistake here, so you'll have to check out the mark scheme. Let's go 250 times 249. I'm going to do them all individually because now, if you're an A-level math student, you're going to be a whiz with your scientific calculator, and you're going to think like, what are some idiots doing if they can't even add up? But if you are someone who doesn't do A-level maths, and you might prefer to do it the real stupid method like mine, 639 200. Let's type that in. So this one equals 639 200. This is the seriously slow method, and you're going to have to catch up in other elements of the paper if you're this bad. But I am doing it so in theory I don't make a mistake. And then 39,800, 39,800. So I've done all of that. Now I just need to add these three numbers together. I'm sure if I just took 5 minutes to actually learn what I'm doing with the calculator, it would be a lot easier than doing it this way. 200 and then plus 39,800, and that should equal 741250. Let's hope that's right, and we haven't made a big 741250. And now I just need to smash this in the calculator and work out what D is. Oh my God, longest 2 marks ever. Divided by 741250. Oh my good gosh, 2.106. So I got D equals 2.106. What we're going to do? Do they say any significant figures? Well, let's give it to 3 peeps. Nah, let's round up there. Yeah, we'll do 3. Okay, so 2.11. That's my index of diversity. Bish, bash, bosh. 2 marks. So I guess any of this working out they see on the paper, and then the correct final answer. Let's go. It's hot, man. It is hot. It's summer, 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 and it is hot. Heat wave coming. 5.4. In a different investigation, the student found out that the species richness of plants in the field of wildflowers is greater than the species richness of plants in a field of wheat. Well, obviously, wildflowers, lots of different species. Wheat, one species, right? Maybe there's a few different species of wheat. The species richness of bees in the field of wildflowers is greater. So there's more different species of bees, different types, honeybee, cuckoo bee, whatever, in the field with the wildflowers than in the field with the wheat. So just one reason why the species richness of bees in a field of wildflowers is greater than the species richness of bees in the wheat field. Well, obviously, if we have a greater species richness in the wildflowers, we can say there are more, oh God, my language is so bad, different species of wild flowers. So there's a greater amount of different species. So more different food sources. So they got more different food sources. Some might have lavender nectar, others might have daffodil nectar, whatever, attract different species of bees. Oh, let's put in lots of different types of nectar because the bees might not all like the same sweet, lovely nectar. They might be like, oh, I like that plant. Oh, I like that plant. So they've got lots. And also more um, habitats. Ooh, more habitats, more shelter, like bigger flowers, enable the bees to hide from predators, things like that. And we could go on and on and on, right? Yeah, so I'll leave it at that. Let's smash that out there. More different types of pollen for reproduction. I don't know. Right, let's zing, let's zing, let's zing. Okay, 6.1, give two roles of phosphate ions in cells. So you know the old phosphate ions, right? Um, Gonna get this wrong, gonna get this wrong. What's a phosphate ion? Is it PO3 4 minus? Wait, that's in group 5 plus 5, that's minus 6. So if I did 3, that would be minus 6 plus 5, that'd be minus 1. So that must be 4 and a 3 minus, oh, you idiot. Right, yeah, that makes sense. Okay, so this is the old phosphate ion. Sorry, just running through my head to work the formula out. If you're a chemist, you'll know phosphates in group 5, so plus 5 charge. Oxygen's group 6 has a minus 2 charge, so you need 4 times 2 to make minus 8. Plus 5 on the phosphate leaves us with an overall 3 minus. That's a phosphate ion. Where do we find this group in biology, in biological molecules? Hopefully, you know, you're going to find it in DNA because you've got the phosphate group attached to the deoxyribose, attached to the nitrogenous base. But we could also say ATP. We could also say, you only need 2 of these. What I'm writing in cap, phospholipids. Yeah, in the bilayer. Yeah. RNA's got phosphates in it as well. Yeah, whatever. Let's move on. Okay, 2 marks. Diseased lungs, I'm not, yeah, whatever, blah, blah. 6.2, diseased lungs can cause carbon dioxide to build up in the blood plasma. This leads to an increase in hydrogen ion concentration in the blood plasma. So we're going to have more hydrogen ions in the blood. Describe, so we don't have to explain, describe the effect this increase in hydrogen ion concentration has on the plasma. So what does an increase in hydrogen ions do to the blood plasma? And what does that in turn do to the proteins in the plasma? So if you want the 2 marks, first of all, say what hydrogen ions do to the blood plasma, and then what the effect on proteins in the plasma is. Do not refer to the Bohr effect. So none of that chloride shift business, HCl3 minus whatever. We don't need to business with any of that. All right, let's get on. So first of all, hopefully you know hydrogen ions make the blood plasma more acidic. Hopefully, if you remember from GCSE chemistry, the higher the concentration of hydrogen ions, the lower the pH. So let's put that as well, lowering the pH. Now, what do we know about pH and proteins, specifically enzymes? What do we know about pH and enzymes? They have an optimum pH, or all proteins do. They're going to lose their tertiary structure if they're in the wrong pH, potentially, and we call that denaturing. So let's say proteins become denatured as, I don't know, their active site changes shape. Now, I could have said proteins such as enzymes, Let's go with such as enzymes. Go on in. Become denatured as their active site changes shape, but it could be just the tertiary structure of any other sort of protein in the blood that loses its shape because of the change in pH. Anyhow, we got the business. Let's just shut the F up, Tom. Right, let's move down. Scientists measured the concentration of inorganic ions in the cytoplasm of mammalian cells in the tissue fluids and the tissue fluids surrounding the cells. So they measured the ions inside the cell and the fluid around the cell. We got the inorganic ion concentration. We got in the cytoplasm, that's inside the cell, and the tissue fluid outside the cell. We've got negatively charged ions, and we've got sodium ions here with the little lines in both. And we've got other positively charged ions. You're thinking potassium, magnesium, etc. Okay. So what are they asking us? Do they want us to explain, use figure 7 to describe two patterns shown in the total concentrations of positively and negatively charged ions in the cytoplasm and in the tissue fluid? So we got some words in bold black here because you may have made a mistake on this. So we're going to go through that. Two patterns they want us to describe in the total concentrations of positive and negative charged ions. Let's do it. Pause the video, have a scribble, unpause, watch me go through it. All right, so first of all, in the total concentrations of positive and negative, I can say that in the cytoplasm, it has the same positive as the number of negative. There's the exact same concentration. And in the tissue fluid, there is the exact same concentration of positive or negative, sorry, as positive. So let's put that down. Remember, it was after the total concentrations. So the same total concentration of positive charged ions and negative charged ions in both cytoplasm and tissue fluid. You could also word this by saying there's no overall charge, which means there's no overall charge, right? So they're both neutral. Neither a positive or overall negative charge because it asked us about the total concentrations. Total concentrations equal no overall charge in the cytoplasm or the tissue fluid. That might be a point we want to make. What am I going to make for my second point? There we go. We got that for 1 mark. Oh, and we're talking about the total concentration. So the total concentration of positive and negative ions is greater in the cytoplasm than the tissue fluid. There we go. That's it. Done. So separate mark, the total conc of ions is greater in cytoplasm than tissue fluid. So I might want to have say the total conc of positive and negative ions in cytoplasm is greater than the total conc of the positive and negative ions. But I've skipped the positive and negative out because from my first statement, the examiner can see what I'm implying. They know I'm talking about both. All right, 2 marks. Let's go. Oh my good gosh. Now, I didn't go over what a misconception here because if you didn't read the bold black carefully, you might have started busting out this stuff and saying, well, in the cytoplasm, it had greater other positively charged ions than in the tissue fluid. Or in the cytoplasm, there were less sodium ions than the amount of sodium ions in the tissue fluid. And you'd be totally correct. Those are accurate descriptions and comparative descriptions. However, they weren't talking about the total concentration of pos and neg. They didn't want you to break it down. All right. So it's a little bit of a trick question, just checking. Part of biology is checking how you can read and understand questions. Okay, 6.4. In these mammalian cells, the cell surface membrane is permeable to sodium ions. Okay, the sodium ion concentration does not increase in the cytoplasm over time, so it stays exactly the same over time. Use this information and figure 7 to suggest and explain the ion transport mechanisms involved in the transport of sodium ions. So what they're saying, let's pretend that my sodium ion conc is, I don't know, 15%, right? Hold on. What the arse am I on about? Let's go up here. Ooh, look, there is a greater amount of sodium ions in the tissue fluid. So let's go down here and let's say that, I don't know, it's 60% sodium in the tissue fluid? I know that might not make sense. Why does it not add up to 100? Don't worry about my numbers, right? Let's get rid of percentage because that might be a bit of a misconception. So I just want to show that there's more sodium here than here. So how must sodium be entering the cytoplasm? Because it says use this information. We know there's more sodium ions outside than inside because we can see from that figure 7. Use this information to suggest and explain the ion transport mechanisms involved in the transport of sodium ions. So how are the sodium ions going in? They're going to be going in by from high to low. So they're going in by diffusion. However, remember our membrane doesn't like to let charged particles through. They have to go through a protein. So what type of diffusion is it? It's facilitated diffusion. So we're going to say sodium ions enter the cell by facilitated diffusion through a channel protein. That's what we're going with. How did we know it's diffusion? Because the graph told us that there was a higher conc of sodium outside the cell than in the cell. Yeah, how do we know it's facilitated? Because they got to go through a protein channel because, of course, they're charged particles. Only non-polar particles like oxygen and carbon dioxide can simply diffuse through a cell. So there we go, bosh. Now, we haven't talked about how it leaves the cell. And the key bit here on how it leaves the cell, it says it does not change or increase over time. So if that's not changing, it's staying 60 and 15, then the same amount of ions that diffuse in are being moved out. And the only way that that can happen is going against the concentration gradient. And what do we call a mechanism that goes against the concentration gradient? We call it active transport. So we're going to say sodium ions are actively transported outside the cell. And that's going to be via a carrier protein or a protein. We can call them pumps, protein pump kind of thing. Okay. And how do we know that's the case? Well, if the sodium ions were just coming in, then the concentration would change. So there has to be the same amount of sodium ions going out as are coming in. We know they're going in by diffusion from high to low. So if they're going out, they've got to be going from low to high. So it had to be active transport. Okay, let's move on. Got a dry mouth, man, and I ain't got no coffee. Did I miss that? Was that the last one? That was the last one. Okay, 7.1. A drug is delivered to decreased. No. A drug is delivered to diseased body cells by a monoclonal antibody. The monoclonal antibody is attached to a drug to form an antibody drug complex, ADC. Got a rectangle here, which is the drug. Complete figure 8 to show a monoclonal antibody attached to the drug. Label one structural feature of the monoclonal antibody in your drawing of the ADC. So ADC stands for antibody drug complex. What they do in biology, hit you with a lot of terms, and they're trying to confuse you. It's just basically draw an antibody attached to this drug. So you got to think about your sketch of an antibody. So we've got that region. God, I can't remember how these look. Is it like this? I think that's it. Is that it? That don't seem right to me. Is the little bit on the outside, should it be like that or on the inside? Do you know what? I don't even know. I'm just going to go like that, and then I'm going to go. Hopefully they accept really bad drawings. Now, the bit where an antibody joins to something else is going to be my constant region. You might want to label this bit over here. What's that? That is going to be the antigen antibody binding site. So antigen antibody binding site. I think there's a load of other things here. Remember, there's 4 polypeptide chains. I'm sure I buggered mine up. And there's supposed to be, isn't there a disulfide bridge somewhere around there? That is the most crap drawing ever. There's a sulfide bridge or disulfide bridge. God, it's the summer. I haven't looked at my textbooks in ages. Disulfide bridge, somewhere around that. Where's the hinge region? Is that the hinge region or the disulfide bridge? Jeez, it's all going pear-shaped here. And then you've got the light chains and the heavy chains, but due to my absolutely crap-shaped Y drawing, let's not get into that. I'm just messing this up. Oh my God. Hold on. This doesn't sit right. I think the heavy chain is like that. And then the light bit is, oh my God, this doesn't sit right at all. Where is the hinge region? Is that the hinge region there? Where's the disulfide bridge? Is that the disulfide bridge? Listen, my memory recall of this is poor at the moment. So you've got four polypeptide chains. If worst comes to the worst, just draw a Y and then write polypeptide chain. They give you the mark. That's definitely the constant region. Antigen antibody binding site. You've also got the variable region, the disulfide bridge, the light polypeptide chains, the heavy polypeptide chains, or just a polypeptide and point to one of the four polypeptides. Okay, right. That'll do. The quicker I get off this question, the better. Right. You only needed to make one mark. Why didn't I just go for the variable region? There we go. Oh my gosh. Scientists use the clinical trial test to Scientists used the clinical trial to test an ADC designed to treat people diagnosed with cancer called Hodgkin lymphoma, HL. In the clinical trial, the patients with HL were selected at random. That's good, avoids bias. Had tumors of a similar size at the start of the trial, standardized. That's good, valid. Received identical doses of ADC. That's very good. Figure 9 shows some of the results from the clinical trial. Figure 9, there was 100 patients with HL treated with ADC. So they've given them all the exact same things and then split it into two groups. I don't get that. But anyway, group 1 has 32 patients. All detectable traces of tumors had disappeared after the course. Group 2 had 40 patients, so they didn't split them. There's 100. Okay, so we've got 32 and 40, and we've got 28 missing. That's a worrying sign. Tumors decreased by more than half the original size for 3 months. So the tumors shrunk for 3 months on average. We don't know what happened after the 3 months, whether they went back to their original size or increased or whatever. Use all the information to evaluate, so good and bad. We can already see some bad things and some good things. The effectiveness of the ADC in treating people with HL. Don't bother mentioning statistical tests. Okay, lovely. So we don't have to write, they didn't do a t-test or whatever. And we're not really comparing these two groups. These are pretty much identical groups because it was 100 people treated with HL. They standardized everything. So that was all really good. If I want to evaluate the effectiveness of this, I would say effective straight away in group 1. And there's probably about 8 things we can say here. So I'm just going to go through 4. In group 1, all 32 patients had no detectable tumors. Now, whether so were cured, it's a bit of a bold statement, isn't it? I would just say they have no detectable tumors. Let's not say they were cured because there might be tumors there that the analytical tests cannot detect. We could mention that maybe as well. We might want to put that as well. And also another good point is that in group 2, the tumors decreased by like 50%. This is also effective in treating HL. So it looks like if I wanted to say, yes, ADC is effective, I might want to start. So this would be my yes, ADC is effective in treating HL. So that's how I might start it. Yes, it's effective. What's my evidence? In group 1, all 32 patients had no detectable tumors. And in group 2, the tumor size decreased by half. This means that all patients, all 72 patients, or I could make a statement here, in these 72 patients, ADL, was it ADC? Oh my days, ADL hash hash. ADC must have had some hash. I'm typing out the wrong thing. In these 72 patients, ADC had a positive effect. Yeah, like on treating the tumors. Great, 2 marks. Now let's go for some bad things. 28 patients' results are missing. So no results for 28 patients. Yeah, they've only given us the results of 32 and 40 and it don't add up to 100. So I'm going to go with no results for 28 patients. Also, for group 2, we do not know if the tumors stayed decreased or returned to original size after jeez, isn't biology painful at times after 3 months? And there's probably a load more things I can say about it. I didn't comment on the sample sizes, about the things they did well, the standardizing, whether some of those are on the mark scheme. I don't know. All right, I'm going to leave it at that because if I keep going on, I'm just going to have to bash my head against the wall. Sometimes, as you draw towards the end of a biology paper, you're like, ugh. Um, a scientist investigated the hydrolysis. Hold on, I missed one. Was that the last one there? Yeah, yeah, yeah. All right. 8.1. A non-competitive inhibitor decreases the rate of an enzyme controlled reaction. Explain how. So, grab this is a nice memory recall question for you. If you don't know the answer, revision card it, guys. Have a go. I'm gonna go through the answer. All right then. So a non-competitive inhibitor means it doesn't compete for the active site. So if this is the active site of my enzyme. Here is my active site. The non-competitive inhibitor um, joins on to a different binding area on the enzyme that's not the active site. There is a keyword you can use called the allosteric site. So I'm going to use that rather than have to say it binds to a part of the enzyme that is not the active site. So it's easier to say the inhibitor binds to an allosteric site. And that is the quick way. It's not on the AQA spec, but you might as well learn it because they accept it. And it means to a different area of the enzyme than its active site. So you might as well say inhibitor binds to an allosteric site. This alters the 3D structure, tertiary structure of the enzyme, um, altering the shape of the active site so it no longer binds or is no longer, I'm going to say is no longer complementary to the active site. So it is no, or let's say, so it can no longer form, because it gets me another keyword in enzyme substrate complexes. Now, these are normally permanent, unlike the competitive. I think that should get me to 3 fat marks. Let's see. It should get me a mark for saying it binds to an allosteric site, alters the 3D shape and changes the shape of the active site, so it can no longer form enzyme substrate complexes. And I might, it does. It already tells us that it decreases the rate of an enzyme controlled reaction. If it hadn't said that, if it had said how does it affect, explain how it affects the enzyme reaction, then I would need to say it decreases it. But I don't need to say that here because it's already said the word decrease there. So I'm just explaining the decrease. Bish, bash, bosh. 8.2, a scientist investigated the hydrolysis of the protein casein. The scientist mixed the solution of protease enzyme with a solution of casein, then measured the casein concentration in the mixture at intervals, controlled all the relevant variables appropriately. For this investigation, identify the independent variable and the dependent variable. Hold on my days. Hydrolysis of the protein casein. They mixed the solution of protease enzyme with a solution of casein, then measured the casein conc in the mixture at intervals. So that's my dependent variable, what you're measuring. Controlled all the relevant variables appropriately. So, oh gosh, that's disgusting. Oh my good God. I don't know what the independent variable is. I know what the dependent variable is. They measured the casein conc in the mixture at intervals, controlled all the relevant variables, i.e. temperature, etc. They mixed the solution of protease enzyme with a solution of casein. All right, so same conc, same volume. Then measured the casein concentration in the mixture at intervals. So the dependent variable is the casein concentration at intervals. I need to look a bit closely because remember, the dependent variable depends on the experiment. So it's not what the scientist does. They measured the concentration of the casein. They can't control the concentration of the casein. How much protein is there is down to the experiment. What the scientists themselves changed, that's the independent variable. So the only thing I can think, which is really hard here for the dependent variable, is that they measured it The scientists chose the interval when we're going to measure it. They chose the time to take the measurement. So the time of the intervals, because it doesn't tell us, oh, they did it every 5 minutes. It tells us they measured it at intervals. So it's up to them when they measured it. That was down to the scientists. They can't control the casein concentration. That's the chemical reaction that's doing that. So that's the dependent. Oh my days, I did not like this. 8.3 shows the scientist results. We've got the casein conc in milligrams per decimeter, not micro. Micro is the U shape. Over time minutes. Use figure 10 to determine the rate of the casein hydrolysis at 2 minutes. Show how you obtained your answer. So you're going to get 1 mark for attempting to do a tangent here, people. I'll go through with it. I'll go through with it. I don't even know what I'm on about. Right, so we're going to go 2 minutes. This is what you're going to do. You're going to draw a line up here at 2 minutes. Look at that bent line. Oh man, come on, Tom, sort it out, mate. Right, oh my days. I'm then going to put a little dot here, and I'm going to remove my little crusty line. Don't need that anymore. So I've gone up there at 2 minutes. I want to draw a tangent. A tangent is a straight line where visually there will be an equal space between the curve and both sides of my straight line. So yeah, I'm rubbish at these on the computer. Okay, I'm going with that, peeps. This probably ain't going to give me the right answer. It looks visually pretty good, right? Roughly equal space either side. I don't know. It looks a bit like I've gone a bit long there. Anyway, now all I need to do is do y over x. So just do y over x. Changing y over x. So going to do y, going to do x. You're going to get a mark for attempting to draw a tangent, even though mine is seriously crusty. I think this line should have come out a bit there, gone in a bit there, but this is as good as I'm going to get it, so I'm going to go for it. What have we got here? That is going to be 1. So my width here is 1 point. How much is each square? Each one of these is 0.2, because there's 2 values divided by 10 squares. What is 2 divided by 10? It's got to be 0.2, right? Oh my gosh, I have to use a calculator for that. I am a real dipstick. So this is at 2. And then what's going up here? That is 10 squares, and we got a value of 50. So 50 divided by 10 is 0.5. So what am I going with here? That is 25. So that's 25. And then 60. So that's 30. So my y apparently is equal to 30. I'm sure people are saying, you idiot, what are you doing? And then this is 1.2. Is that 1.4 or is it 1.45? See, the 1.45 minutes. Is it 1.5 minutes? Hold on. 1.5 minutes. Wait, that's 3. I'm a real dipstick. This is either 1, 2, 3, 4, 5. Oh my days. So that's 1 minute, 1.2, 1.4, 1.6. Oh my gosh. What is X? What is X? I don't know. Shall I go 1.5? 1.6? I'm going 1.5. All right. And then bish bash bosh. Let's have it. Probably completely wrong. 30 divided by 1.5. So I have got the rate of casein hydrolysis is 20 milligram decimeters. They're probably giving me the units. Does it want the units? And then min minus 1. So yeah. I hope they gave us the units. So milligram decimeter min minus 1. Yep. I'm going 20. It's probably not 20. Okay, I'm going with that peak. You have to check the mark scheme. I think 20 will be acceptable on the mark scheme, but obviously when I'm doing on the computer, my accuracy is limited, blah, 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 excuses. 8.4, the scientists repeated the investigation but increased the temp to the optimum temperature for this protease, so they used the optimum temp. Sketch a line showing the results. You predict if the investigation is repeated at the optimum temp for protease. Okay, let's sing up. So we're using a better temperature, so the casing is going to break down faster. So we want to start in the same point, and the line's just got to be less, right? So it's going to come down faster at the optimum temperature. Does it reach zero? Yeah, it reaches zero. So we're just going to get there faster. That's all. So, oh my gosh, that is some crusty line. So I've just gone like that. Bash. All right, and there we go. Obviously not as bent as my line, but one can't help it. Was that one mark? That's got to be one mark. It's 2 marks. Bloody hell. There we go. Right. I'm so tired. No. Read the following passage. DNA is a stable molecule, but even under normal cell conditions, spontaneous changes occur to the DNA nucleotide sequence. One example of a spontaneous change occurs when a cytosine base in guanine cytosine nucleotide pair is changed to a uracil base. This produces a guanine uracil nucleotide pair in the DNA molecule. Scientists estimate this type of spontaneous change occurs to 100 guanine cytosine nucleotide pairs in the genome of the healthy human cells every day. In healthy humans, no, in healthy cells, enzyme control processes repair these spontaneous changes in the DNA molecule by changing uracil bases back to cytosine bases. If these repairs do not happen, the uracil DNA nucleotide attracts an adenine DNA nucleotide. When the DNA is replicated in the cell cycle, a mutation of the original DNA has now occurred. Healthy cells with damaged DNA produce enzyme X. This enzyme slows down the cell cycle by delaying the start of DNA replication. People with the disease ataxia telangiectasia, can't say it, AT, thanks for that, do not produce functional enzyme X. Mutations occur at a higher rate in people with AT. Use the information in this passage and your own knowledge to answer the following questions. 9.1. Give one similarity in the structure between guanine cytosine nucleotide pair and a guanine uracil nucleotide pair. In a DNA molecule, we actually don't need to have read any of the passage for this. So do not refer to guanine. So they're saying, what is a similarity between a guanine cytosine and a guanine uracil nucleotide pair? So are they adjacent or are they opposite each other? So they're held together, a similarity. They're both held together by hydrogen bonds. And both the cytosine nucleoside and the uracil nucleoside contain a phosphate group. Maybe we could say that as well. I don't know whether they'll accept that, but let's go for it. Uracil and the cytosine both contain a phosphate group. Um, we don't want to start bussing out deoxyribose because one's ribose and one's deoxyribose, right? Potentially the uracil nucleotide pair. In the uracil, in the guanine uracil nucleotide pair. So I think they're both going to contain phosphate, whether they're going to allow us to have that. Both contain a nitrogenous base, both are nitrogenous bases. They probably won't allow that. They're both held together by hydrogen bonds, aren't they? The guanine to cytosine and the guanine to uracil, they're both hydrogen bonds. All right, let's zing a mark there and move on. All right, waffle so much. This is like a 2 hour walkthrough. It's disgusting. I'm going to go back to my old style. 9.2, the DNA in a human genome contained 3 times 10 to the 9 nucleotide pairs. Assume 40% of these nucleotide pairs are guanine cytosine nucleotide pairs. Use this information in lines 5 to 7 to calculate the percentage of guanine cytosine nucleotide pairs that change to guanine uracil nucleotide pairs in the genome every day. Give your answer in standard form. Show your work in 2 marks. Let me get this straight. So what we're doing here, we're saying that 3 times 10 to the 9 nucleotide pairs in total in a human genome. Right. 40% of these are GC. So let's work out how many GC pairs in the human genome. So we're going to go 0.4 times 3 times 10 to the 9, and that equals 12. So we're going to go 0.4, because that's 40%, right? 40 divided by 100, whatever, times 3 times 10 to the 9. This gives me a total GC pairs My total GC pairs in the human genome is, what the hell is that? Three. Okay, that's a lot. Yeah, that's a lot. 100. What's that? 1,200 million. That's 1.2 billion or something. Right, there's a lot of GC pairs, 1.2 billion or something in the human genome. Not really sure how much that is. It's a big number, 12 with eight zeros. What I need to do is go up here. There was something up here. It said line 7. This produces a guanine uracil nucleotide pair in the DNA. Scientists estimate, here we go, line 6, line 7. It occurs to 100 GC nucleotide pairs every day. So 100 of these bad boys are going to mutate. So what's that as a percentage? So I want to do 100 over that. Oh, it's not so bad, actually, this one. Normally biology ones are flipping disgusting. And then times 100. So I'm just going to put 100 divided by my answer equals this times 100. So we are looking at 8.3 times 8.33 times 10 to the minus 6%. 8.33 times 10 to the minus 6% of mutations in these GC pairs that then are turned into G uracil. I have to be honest, that's a tiny amount. How amazing is the human DNA's replication process? You couldn't get that small error in any factory or industry when they're making stuff. You know, like if you buy a Tesla, you buy one of these Casio calculators, there's going to be a certain percentage that are faulty that you buy out the packet. It's not going to be as small as that. That is highly effective. Human body's incredible, people. As if that's the right answer. Oh, 9.3. Please tell me that's the end. Oh my gosh, 7 more marks. This has been the longest walkthrough ever. I do apologize. I'm going mad now. The type of mutation that occurs when the repairs do not happen, lines 10 and 11, may still produce a functional protein. Okay, so even when the repairs don't happen, you still might get a functional protein. Explain why. What is the funky thing about DNA? What do we want to waffle on about here? Think about DNA's properties. So hopefully you got your revision cards out. If you don't know this, remember the properties of DNA. It's non-overlapping. It's universal. The same um, base codes, the same 4 bases code for the same amino acids in all living organisms. But the one we're looking at here is DNA is a degenerate code. It means that there is more than one codon that codes for each amino acid. So this may be a substitution or a silent mutation, or it is a substitution and it could be a silent mutation. So we might want to say DNA is degenerate. I can't spell degenerate. DNA is degenerate like me. This means more than one codon codes for each amino acid. I'm going to change that word codon just in case I get myself in trouble. More than one triplet codes for each amino acid. It is a substitution mutation because we've swapped C for U, right? Guanine, cytosine to guanine uracil. So it's substitution mutation. So it could be silent and code for the same amino acid, even though the, oh my God, triplet code has changed. This means, I'm longing this one out, but this means the protein, functional protein, this means the protein will have the same primary structure, or so you can have the same primary structure, the same order of amino acids. So the same primary structure, again, sequence of amino acids. That's the same thing as saying same primary structure. I've kind of said it twice. So same sequence of amino acids. So it's going to have the same hydrogen bonding secondary structure, but more important, so same tertiary structure, same ionic bonds, covalent bonds, etc. So the same shape, 3D shape. I've longed this out, but I've covered all bases. So we've gone DNA's degenerate. This means that more than one triplet code codes for each amino acid. This is a substitution mutation because cytosine swap for uracil. So this means that it might be a silent mutation, in which case we may get the same amino acid coded for, even though the triplet code has changed. This means the protein's going to have the same amino acid sequence because the amino acids are the same, which means that we're going to get the same folding in the secondary structure, and we're going to get the same ionic bonds, covalent bonds, hydrophobic, hydrophilic interactions in the tertiary structure to give us the same 3D shape and therefore the same function, i.e. if it's an enzyme, it will still be complementary to the substrate, still form an enzyme substrate complex. Bish bash bosh, 4 marks. Let's go. Suggest and explain why mutations occur at a higher rate in people with AT. I'm done, folks, and there's 3 more marks. Let's do it. Let's do it. Lines 15 and 16. After all of that, reading that long ass passage, we hardly needed it. So people with the disease let's just call it AT, ataxia telangiectasia. I can't, I'll have to like practice that. I can't speak that. People with that disease, AT, do not produce the functional enzyme X. Mutations occur at a higher rate with people with AT. So we can presume that the enzyme X, which is said here, controls the repair these spontaneous changes. I hate these people, man. It said lines 15 and 16. All it says in lines 15 and 16 is that they do not produce enzyme X. This doesn't help you, but if we go back up here, in healthy cells, enzymes control processes repair these spontaneous changes. Nucleotide attracts an adenine nucleotide is a replicating cell cycle and mutation original. Healthy cells with damaged DNA produce enzyme X. So that's lines 13. This enzyme slows down the cell cycle. So we really needed line 13 or 14, not 15 and 16. So we can go to our answer now. We know that enzyme X slows it down. So it says here, people with AT do not produce enzyme X. So they do not produce enzyme X. This means the cell cycle is not slowed. Come on, Tom, slowed down. So it occurs. Oh my days, man, I'm tired. At a higher rate. So a higher frequency of mutations occur. I reckon that's it, right? So they don't produce enzyme X is going to be 1 mark. This means the cell cycle happens faster because it doesn't slow down, so it goes at a faster rate. So we're going to get more mutations per second. Yeah. Bosh! Don't forget sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks. I'll check you guys in the next one.
You're still here? It's over.